In 1964, the identification of the primary psychoactive compound in cannabis, THC, opened the door to a new field of medical research. The exploration of the therapeutic potential of THC and other natural cannabinoid compounds came some years later together with the discovery of the endocannabinoid system. But what are endocannabinoids?
Endocannabinoids are derived from fatty acid, and it's curious to see that already lower plants such as mosses, ferns, algae also, but also fungi, they do contain endocannabinoids, so they invented them in evolution almost, and then later when it came to animals, endocannabinoids became important for the immune system and in the brain. And that's why we are interested in them. In the brain, they're major neuromodulators, and in the immune system, they're major immunomodulators. And that's why they're also relevant for diseases. One aspect that's not yet understood within the endocannabinoid system is how these molecules, these endocannabinoids, are transported across the cell membranes and how they're then degraded. And within NCCR TransCure, we dedicate a lot of time to understand this mechanism. And we have been developing compounds that selectively and potently target this process. And we're also interested in elucidating the molecular mechanism of this process. So once again, we got inspired by natural products for our project. We discovered the first endocannabinoid reuptake inhibitors in echinacea, that's the corn flower. And from that we then generated the semi-synthetic libraries and further modified the molecule so that in the end we got a very potent and selective synthetic molecule. We also work with purely synthetic molecules such as this one. So these molecules are profiled in vitro, but also in vivo in animal models. And what we are ultimately interested in is whether these molecules are bioavailable to the brain, whether they exert pharmacologically relevant effects in certain types of diseases.
So we use several biochemical assays to profile the mechanism of action and the selectivity and potency of compounds that can modulate endocannabinoid transport.
Selective endocannabinoid reuptake inhibitors are a new class of molecules that they show a new mechanism of action that modulates the endocannabinoid system. They act as inhibitor of the reuptake, cellular reuptake of endocannabinoids, and they show therapeutic effect in vivo. We have now selected some potential lead candidates that they show no cytotoxicity, no acute toxicity in vivo, and good physicochemical properties. In line with the mechanism of action, analytical studies show that selective endocannabinoid reuptake inhibitors increase the level of endocannabinoids in immune cells, in neurons, and also in vivo.
We are of course interested in the end to see whether our mechanism that we discover also would work in humans, and whether also the diseases that are relevant to humans could be treated eventually with our compounds, and that makes it quite interesting.